Hi, my name is Aijing Feng. The topic I'm going to present today is genomic diversity and associates phenotyping of E. coli isolates from Poultry in the southern United States. So, avian pathogenic E. coli is one of the most common disease-caused bacteria. After E. coli enter through oral, nasal, cloacal routes, APEC colonized the mucosal sites of the GI tract, respiratory and reproductive tracts, and results in localized and systematic infects that threaten animal welfare and the production in domestic poultry. Antibiotics are used but bring the emergence and re-emergence of biological resistant bacterial strains and some potential untreatable bacteria, which in the end threats both human and animal health. An effective vaccination program is an important strategy in E. coli disease prevention and control for the alternative to antibiotics, which requires to understand the underlying mechanism of pathogenic E. coli. Identifying the genes encoding one or more variance factors in pathogenicity islands and genes that provide bacterial resistance to antibiotics in resistant island is one of the ways. However, study shows that the subset genes involve different functions such as the bacterial adhesion, invasion, toxicity, antibiotic resistance, survival, and metabolism under stress instead of single genes, plasmid, phase, or pathogenicity island could well associate with the APEC disease. So the project goal of this study aimed to explore the E. coli isolates genomic diversity with their associated phenotyping of the infection locations. And we hypothesized that a subset of genes from the whole genomic sequence could associate with disease in different infection locations. And then those selected subsets of genes would include different functions, and some of them may be PAI or the REI. 187 E. coli strains were isolated from the sick poultry samples between May 2017 and July 2021. And those isolates cover diversity in the host species, age, and infection locations, including those important infection locations I mentioned above. Phylogenetic analysis was conducted using the CV tree, and large genetic variation was shown in the whole genomic tree, and 16S rRNA genes were clustered into two major groups. There are 5,069 genes identified by the annotation software, and about 1,100 were sharing genes through all the samples. The rest 3,933 genes were used to build the gene matrix. And one group of the gene matrix described the presence and the absence of the genes in the samples using 1 and 0. The other group of the gene matrix described the gene duplication with numbers. And the infection locations were encoded as one hot matrix. The multitask feature learning models could be used to learn the features from this genetic matrix and the phenotype matrix. The model I use for this study is the tree guide multitask sparse feature learning on the logistic regression model. So the numbers of the gene features is larger than the numbers of samples. Numbers of the sample in each task is insufficient to train an accurate learner, while giant learning multiple related tasks so that the knowledge contained in a task can be leveraged by other tasks with the hope of improving the generalization performance of all the tasks. The tree structure was used to group the gene features based on the prior knowledge. Regularization is used to force some of the genes' weights to be zero during the model learning for feature selections. And the bootstrapping resembling is also used for the robust feature selection. The final object function is showing in the bottom of the page. 27 genes were selected in total with prediction accuracy of 87% in the test set. All the selected genes were in the gene duplication group, and the gene functions were identified using Python software. It includes 12 different pattern family and subfamily, and 5 different protein classes. Those selected genes also include 4 variance factors, and 2 of them are PAI. So this is the summary of this study. So 187 E. coli strains isolated from the sick poultry samples were collected and sequenced. Samples cover diversity in the host species and age and the infection locations, as well as the large genomic diversity. We encode the genetic features and the infection locations. The tree guide multitask logistic regression model was used for the sparse feature learning, and 27 genes were selected in total with 87% prediction accuracy in the text set. The selected features include different protein functions and 4 variance factors, and 2 of them are PAI. This study explored the genes associated with different infection locations based on the lab collected 187 samples. Published data set could be added to explore and selected disease-related genomic features. The variation within specific genes, variations in nucleotide, and the hypothetical proteins, non-coding regions in the sequence could be considered. I would like to thank the colleagues from USDA and the university, the Mississippi State University, and also thank the grant support from the USDA. Thank you.